Fortuitously, the Wellcome Trust's Michael Morgan was able to give the public project a welcome boost just days after Venter's announcement by declaring that it would be doubling its support for the Sanger Center, bringing the total up to around $350 million. Though the timing of the announcement made this look like a direct response to Venter's challenge, the increase in funding had, in fact, been in the works for quite some time. Shortly afterwards, the U.S. Congress beefed up its own contribution to the public HGP's coffers. The race was on. In fact, from the outset, there were always going to be at least two winners. Science only stood to benefit from two human genome sequences, one against which to check the other. With over 3 billion base pairs involved, there was bound to be a typo or two. Another winner would surely be ABI. They stood to sell a lot more PRISM sequencing machines, which most labs in the public project would now have to buy to keep up with Venter. The acrimonious exchanges between the leaders of the private and public projects would become a fixture of newspaper science pages for the next couple of years. The back and forth got to a pitch that moved President Clinton to direct his science advisor, "Fix it. Make these guys work together." But through it all, the sequencing moved ahead. And Venter did demonstrate that a WGS approach could work on a respectably sized genome when, in collaboration with the fruit fly wing of the public consortium, he announced the completion of an advanced draft of the Drosophila genome early in 2000. It, however, contains relatively little repetitive junk DNA, and Celera's success in assembling it in no way guaranteed WGS would work on the human genome. No individual was more vital to meeting the Celera challenge than Eric Lander. It was he who envisioned an almost entirely automated sequencing process in which robots would take the place of technicians, and it was he who had the drive to make this vision a reality. Lander's resume indeed shows he knew a thing or two about drive. A Brooklyn boy, he was a curve-busting math whiz at Stuyvesant High in Manhattan, who went on to win first prize in the Westinghouse Talent Search. He then became valedictorian of his class at Princeton, 78, before earning his Ph.D. at Oxford on a Rhodes Fellowship. A MacArthur Genius Award in 1987 seemed almost redundant. His mother, incidentally, has no idea how it all happened. I'd love to say I'm responsible, but it's not true. I'd have to say it was dumb luck. Ultimately finding pure mathematics an isolated monastic kind of field, Lander, notably gregarious by the standards of his discipline, joined the jollier faculty of the Harvard Business School. But he soon found himself distracted and intrigued by the labors of his younger brother, a neuroscientist. Inspired, Lander taught himself biology by moonlighting in Harvard's and MIT's biology departments, all the while scarcely missing a beat on his day job at the B School. "I pretty much picked up molecular biology on street corners," he says. "But around here, there are a lot of very good street corners." In 1989, he became a professor of biology on one of those street corners, MIT's Whitehead Institute. Even among the so-called G5, the public effort's 5 major centers, which also included the Sanger Center, Washington University's Genome Sequencing Center, Baylor College of Medicine, and the DOE in Walnut Creek, Lander's lab would be the largest single contributor of DNA sequences. His team at MIT would also be responsible for much of the enormous acceleration of productivity in the home stretch leading up to the release of the draft genome. On November 17, 1999, the public project celebrated its billionth base pair with the sequencing of a G. Just 4 months later, on March 9, 2000, a T was base number 2 billion. The G5 was cranking. Because Celera was using the public project's data, which were posted immediately on the internet and were now pouring in thick and fast, Venter, perhaps finally breaking a sweat, halved the amount of sequencing he had originally projected Celera would do. As the public-private race reached a climax in the media behind the barricades, the focus was increasingly shifting to the effort's mathematical brain trust, scientists hidden in back rooms among banks of computers. They were the ones who had to make sense of all those A's, T's, G's, and C's of crude sequence. They had two major tasks. First, to assemble a whole final sequence from the many, many discrete chunks on hand. Most parts had been sequenced numerous times, so there were several genomes worth of sequence to sort out, all of which had to be distilled down into a single canonical genome sequence. Computationally, this was an enormous undertaking. Second, to figure out what was what in the final sequence, and, above all, where the genes were. Identifying the genome's components, distinguishing between one stretch of A's, T's, G's, and C's that encoded nothing but junk, and another that encoded a protein, depended on extremely computer-intensive approaches. At the heart of Celera's computer operations was Gene Myers, the computer scientist who had been the WGS approach's first and most forceful advocate. With James Weber of the Marshfield Medical Research Foundation in Wisconsin, he had proposed that the public effort adopt WGS long before Celera even came into existence. And so for Myers, the success of Celera's bid was a point of pride and vindication. Anchored as it was by previously mapped genetic landmarks, the public project's job in assembling the sequence, though immense, seemed less daunting than the one confronting Myers in the landmark-free world of WGS. In its final analysis, Celera used the public project's freely available map information. In fact, in counting on these very landmarks, the public project had rather underestimated its own computational challenge. So as Celera added computer muscle, the public project stayed focused on gearing up its sequencing operation. Only very late in the day did the leaders of the public project realize that despite the map, they too, like the proverbial father facing the parts of the new bike on Christmas Eve, had a major assembly problem on their hands. A date for completion and assembly of the rough draft had been fixed for the end of June. But at the beginning of May, the public project still had no working means of assembling all their sequences. Their deus ex machina took a strange form, a graduate student from UC Santa Cruz. His name was Jim Kent, and he looked like a member of the Grateful Dead. He had been programming computers since the beginning of the PC era, writing code for graphics and animations, but then decided on graduate school so he could be part of bioinformatics, the new field dedicated to analyzing DNA and protein sequences.